Welcome to Softly Curious. Tonight, we begin with structure, not history, not medicine, not the future. Just form, the quiet physical design of DNA itself. Inside nearly every cell, DNA exists as a long, continuous molecule. If fully stretched, the DNA inside a single human cell would measure close to 2 meters in length. Yet it fits within a nucleus far smaller than a grain of sand. That compression is remarkable, but before folding and packaging, there is the shape itself. DNA forms what is known as a double helix. A helix is a spiral structure, like a gently twisting staircase. In DNA, 2 strands wind around each other in a steady, repeating pattern. This geometry is not decorative, it is precise. Each turn of the helix spans about 10 base pairs, and the diameter remains consistent along its length. The symmetry gives the molecule stability. The twist gives it strength. The 2 strands do not run in the same direction. They are anti-parallel, meaning one strand runs in a 5 to 3 direction, while the other runs 3 to 5. These numbers refer to carbon positions within the sugar molecule that forms part of DNA's backbone. Anti-parallel orientation allows the bases to align properly across the center. Without this opposite alignment, the pairing would not hold. Each strand is built from repeating units called nucleotides. A nucleotide is a small molecular unit made of 3 components: a sugar, a phosphate group, and a nitrogen-containing base. In DNA, the sugar is deoxyribose. The term deoxyribose simply means ribose lacking 1 oxygen atom. That missing oxygen slightly changes the molecule's chemical behavior, making DNA more stable than its close relative RNA. The sugar and phosphate groups connect to form what is known as the backbone. Phosphate groups link the sugars together through phosphodiester bonds, creating a repeating structural chain. This backbone sits on the outside of the helix. It provides rigidity and protects the more delicate base pairs inside. If you imagine the double helix as a ladder, the backbone forms the sides, while the bases form the rungs. The analogy works briefly, but the actual structure is more refined. The rungs are not identical bars. They are specific chemical pairings. There are 4 primary bases in DNA: adenine, thymine, guanine, and cytosine. Adenine pairs only with thymine. Guanine pairs only with cytosine. These pairings follow strict rules because of their molecular shapes and bonding patterns. Adenine and thymine connect through 2 hydrogen bonds. Guanine and cytosine connect through 3. Hydrogen bonds are relatively weak interactions compared to covalent bonds, but in large numbers, they provide significant stability. The difference between 2 and 3 hydrogen bonds may seem small, but it affects the overall strength of the helix. Regions rich in guanine and cytosine are slightly more stable because 3 bonds hold each pair together. This subtle chemical variation influences how tightly certain regions remain bound. Beyond hydrogen bonding, another stabilizing force operates quietly along the length of DNA. This is base stacking. Base stacking refers to the way adjacent bases align and interact through shared electronic forces. The flat surfaces of the bases stack on top of one another, creating stabilizing interactions along the axis of the helix. In many ways, stacking contributes more to overall stability than the hydrogen bonds themselves. The geometry of the helix creates 2 grooves along its surface. These are known as the major groove and the minor groove. They form because the base pairs are not centered exactly between the 2 backbones. One side opens slightly wider than the other. The major groove is wider and exposes more chemical information from the base pairs. The minor groove is narrower and reveals less detail. Proteins that interact with DNA often recognize specific sequences by fitting into the major groove, where more of the molecular pattern is accessible. The grooves act like subtle contours, guiding molecular recognition without disrupting the helix itself. The form most commonly found in living cells is known as bDNA. bDNA refers to the standard double helical structure described in early models. It has a right-handed twist, meaning it spirals upward in a clockwise direction. Its dimensions are consistent, its grooves predictable, and its stability reliable under physiological conditions. The model of this structure became widely known in 1953 when James Watson and Francis Crick proposed the double helix arrangement. Their model was informed by X-ray diffraction data, particularly an image known as Photo 51, produced through the work of Rosalind Franklin and Morris Wilkins. The X-shaped pattern in that image revealed the helical nature of DNA with striking clarity. The structure was not guessed. It was inferred from physical evidence. What made the double helix extraordinary was not only its appearance, but its logic. The pairing rules immediately suggested a copying mechanism. If one strand carried a sequence, the complementary strand inherently carried the matching information. Structure implied function without needing to state it explicitly. But for now, we remain with structure. The diameter of bDNA remains close to 2 nanometers across. Each full turn measures about 3.4 nanometers in length. These measurements repeat with remarkable consistency. DNA is not chaotic. It is geometrically disciplined. The twist of the helix introduces tension. It allows the molecule to coil and uncoil without breaking. The hydrogen bonds between bases are strong enough to hold the strands together under normal conditions, yet weak enough to separate when necessary. This balance between stability and flexibility is built directly into the architecture. At a molecular scale, the helix is not rigid like metal. It bends slightly. It breathes. Individual base pairs can momentarily separate and reconnect. These tiny fluctuations occur constantly due to thermal motion. The structure is stable, but not frozen. If you could shrink down to its scale, you would not see a static sculpture. You would see a gently vibrating form, maintaining its pattern through motion rather than stillness. Even the anti-parallel orientation contributes to mechanical integrity. By running in opposite directions, the strands distribute torsional stress more evenly along the helix. The backbone's negative charge, carried by phosphate groups, also influences interactions with surrounding molecules and ions inside the cell. All of this arises from repeating simplicity: sugar, phosphate, base, over and over, billions of times. The elegance of DNA's architecture does not come from complexity at the level of parts. It comes from repetition organized with precision. 4 bases, 1 sugar type, 1 backbone pattern, a twist maintained across immense length. Within that simplicity, information can vary endlessly. The order of bases changes, but the geometry does not. The form holds steady while the sequence shifts. In bDNA, the dominant form inside cells, this balance between uniform structure and variable content allows the molecule to function as both scaffold and script. Its shape protects the information it carries. Its pairing rules preserve that information during copying. Its grooves expose just enough detail for recognition without compromising stability. DNA's physical design is not accidental ornamentation. It is an arrangement that allows length without fragility, stability without rigidity, and repetition without monotony. At its core, the double helix is a structural solution to a fundamental challenge: how to store immense amounts of information inside a space too small to see, while keeping that information accessible and durable across time. For now, we remain with the shape itself. 2 strands, winding, a quiet symmetry, a structure that holds steady, even as the life built upon it changes constantly. DNA is the molecule that stores genetic information in living organisms. It carries instructions in the form of chemical sequences, allowing cells to build proteins, regulate activity, and pass traits from one generation to the next. But for DNA to do this reliably, it must not only store information. It must endure movement, pressure, bending, and constant molecular traffic inside the cell. Its shape, therefore, is not fixed in a single rigid form. It adapts. Although bDNA is the dominant structure inside cells, it is not the only one DNA can adopt. Under different environmental conditions, such as changes in hydration or ionic concentration, DNA can shift into alternative conformations. One of these is aDNA, a slightly shorter and wider version of the helix. In aDNA, the base pairs tilt more noticeably and the grooves change proportion. This form appears more often in dehydrated conditions and in certain DNA-RNA hybrid regions. The sequence remains the same, but the geometry subtly shifts. Another structural variant is zDNA. Unlike the familiar right-handed spiral of bDNA, zDNA twists in a left-handed direction. Its backbone follows a zigzag pattern, which is where the letter Z comes from. This structure was studied extensively by Alexander Rich, who demonstrated that DNA is capable of reversing its helical direction under specific conditions. zDNA tends to form in regions rich in alternating guanine and cytosine bases. It is less common, but its existence reveals that DNA is structurally flexible at a fundamental level. Beyond these alternative helical forms, DNA also experiences higher-order twisting known as supercoiling. Supercoiling occurs when the helix is twisted upon itself, adding extra turns or removing turns from the standard helical winding. If bDNA is like a gently coiled rope, supercoiled DNA resembles a rope that has been twisted further until it loops back on itself. Supercoiling can be negative or positive. Negative supercoiling occurs when the DNA is slightly underwound, meaning it has fewer helical turns than usual. Positive supercoiling occurs when it is overwound, containing more turns than its relaxed state. These differences influence how easily the strands separate. Negative supercoiling can make strand separation slightly easier, while positive supercoiling increases tension. This torsional stress, meaning twisting strain along the axis of the molecule, is not accidental. It emerges naturally as DNA is replicated, transcribed, and packed. The molecule behaves less like a rigid rod and more like a flexible cable that responds to torque. It absorbs and redistributes mechanical stress rather than snapping under pressure. DNA is also capable of forming local structural features beyond the double helix. One example is the Guy quadruplex. This structure forms in guanine-rich regions where 4 guanine bases arrange themselves into a square planar, configuration stabilized by specific hydrogen bonding patterns. Instead of the standard base pairing between 2 strands, G quadruplexes create stacked layers of 4 base units. These regions are often found near gene regulatory areas and telomeres. Another less common configuration is triplex DNA. In this arrangement, a 3rd strand binds to the major groove of a standard double helix through additional hydrogen bonds. The result is a 3-stranded structure, stabilized under particular sequence conditions. While not widespread across the genome, triplex formation shows that DNA's chemistry allows more than simple pairing. Its grooves provide access points for additional molecular interactions. All of these structural variations are possible because DNA is elastic. Elasticity refers to its ability to bend and return to its original shape. At short-length scales, DNA can curve smoothly. At longer scales, it can loop, fold, and twist without breaking its backbone. This elasticity is essential inside the cell, where DNA is constantly being compacted and rearranged. Inside the nucleus, DNA exists at an extraordinary density. Nearly 2 meters of DNA are confined within a space only a few micrometers in diameter. This level of compaction creates molecular crowding. Proteins, ions, and other macromolecules surround the DNA continuously. The helix must remain stable in this crowded environment, while still allowing access where needed. Under such dense conditions, mechanical forces become significant. Molecular motors that move along DNA generate localized tension. As they progress, they introduce additional twisting ahead of them and relieve tension behind. The DNA responds dynamically, adjusting its supercoiling and distributing stress across nearby regions. The molecule is not passive. It behaves like a responsive structure, maintaining integrity while accommodating force. Taken together, these structural variants and physical properties show that DNA is not a static blueprint. It is a dynamic polymer, meaning a long repeating molecular chain, capable of shifting form in response to its environment. Whether adopting aDNA or zDNA, forming quadruplex regions, or absorbing torsional strain through supercoiling, the molecule demonstrates a balance between stability and adaptability. Its architecture provides continuity. Its flexibility provides resilience. And within the confined, crowded interior of a living cell, that combination allows DNA to endure as both structure and system, quietly holding its shape while constantly adjusting to the forces around it. Inside the nucleus, DNA does not float freely. It cannot. Nearly 2 meters of genetic material must fit into a space only a few micrometers wide. If stretched out, the DNA from a single cell would extend far beyond the cell itself. Yet in reality, it remains compact, organized, and accessible. This remarkable compression is not random folding. It is a carefully structured system. The first level of packaging begins with histone proteins. Histones are small, positively charged proteins that bind to the negatively charged DNA backbone. Because DNA carries a negative charge due to its phosphate groups, it is naturally attracted to these positively charged proteins. This electrostatic attraction allows DNA to wrap around histones securely. 8 histone proteins assemble into a core structure known as a histone octamer. Around this core, approximately 147 base pairs of DNA wrap in about 1.5 turns. The result is called a nucleosome. A nucleosome is the fundamental unit of chromatin, meaning the combined complex of DNA and protein that makes up chromosomes. If you imagine a long thread, the nucleosomes would appear like evenly spaced beads along it. Each bead represents DNA wrapped around histones. The thread connecting them is known as linker DNA. This repeating arrangement reduces the effective length of DNA significantly, while maintaining an ordered structure. But nucleosomes are not static spools. They can shift slightly along the DNA, loosen their grip, or tighten depending on cellular conditions. This flexibility allows certain regions of DNA to become more accessible when needed. As nucleosomes interact with one another, they fold into higher-order structures. These folds create chromatin fibers, which then loop and coil further to form the familiar shape of chromosomes. The process is hierarchical. DNA wraps around histones. Nucleosomes assemble into fibers. Fibers fold into loops. Loops compact into chromosomes. Chromatin does not exist in a single uniform state. It appears in 2 general forms, euchromatin and heterochromatin. Euchromatin is less densely packed and tends to be more accessible. Genes located in euchromatin are often actively used by the cell. Heterochromatin, by contrast, is more tightly packed and generally less active. The distinction is not absolute, but the density of packing strongly influences gene availability. Under a microscope, these differences become visible. Tightly packed heterochromatin stains more darkly, while euchromatin appears lighter. The visual contrast reflects underlying physical organization. In dense regions, DNA is wound more tightly around histones and folded into compact domains. In open regions, the structure is looser. The ends of linear chromosomes are protected by structures called telomeres. Telomeres consist of repetitive DNA sequences that do not encode proteins but serve as protective caps. Each time a cell divides, a small portion of the telomere sequence is lost due to the mechanics of DNA replication. Over time, telomeres gradually shorten. Elizabeth Blackburn contributed significantly to understanding telomeres and the enzyme telomerase, which can extend them. Telomerase adds repeated sequences to chromosome ends, helping maintain stability in certain cell types. Without telomeres, chromosome ends could be mistaken for broken DNA, triggering repair systems that might fuse chromosomes together. Near the center of each chromosome lies the centromere. The centromere is a specialized region that plays a critical role during cell division. It serves as the attachment site for spindle fibers, which pull chromosomes apart into daughter cells. The centromere is not defined solely by its DNA sequence. Its identity is also influenced by specific proteins and structural features. The linear chromosome system found in humans and many other organisms allows DNA to be divided into distinct units. Humans have 23 pairs of chromosomes, each containing a continuous DNA molecule. These chromosomes vary in length and gene density, but each follows the same fundamental packaging logic. The organization of chromosomes inside the nucleus is not chaotic. Individual chromosomes occupy distinct regions known as chromosome territories. They do not tangle randomly. Instead, they maintain relative positions that influence how genes interact and how regulatory elements reach their targets. The idea that chromosomes could change position or structure was explored by scientists such as Barbara McClintock. Through her work on maize, she discovered that certain DNA elements could move within the genome. These mobile elements, later called transposons, revealed that the genome is not entirely fixed. Even within its packaged form, DNA retains the capacity for rearrangement. Packaging is therefore not simply compression. It is regulation, protection, and spatial organization combined. The cell must strike a balance. DNA must be compact enough to fit within the nucleus, yet accessible enough for transcription and replication. Mechanical stability also matters. During cell division, chromosomes condense even further into tightly coiled structures visible under a light microscope. This condensation prevents tangling and breakage as chromosomes are separated. Once division is complete, the structure relaxes back into less condensed chromatin. At every level, from nucleosome to chromosome territory, packaging shapes function. Regions wrapped tightly are generally quieter. Regions that loosen become active. The physical arrangement influences which genes are read and when. Despite this complexity, the underlying principle remains straightforward. DNA is folded in layers. Each layer adds stability while preserving order. The genome becomes not just a long molecule, but an organized architecture within the nucleus. In the confined interior of the cell, structure and accessibility must coexist. Histones hold DNA in place. Nucleosomes define repeating units. Chromatin fibers fold into domains. Telomeres protect the ends. Centromeres guide separation. Linear chromosomes preserve continuity across generations. The result is a system that transforms an immense molecular length into a compact, stable, and functional form. DNA does not simply reside inside the nucleus. It is arranged, anchored, and structured with deliberate precision. And within that structured quiet, information waits folded, protected, and ready to be unfolded when the cell requires it. Not all genomes are arranged as long linear chromosomes inside a nucleus. While humans and many other organisms rely on linear DNA packaged into distinct chromosomes, life has developed additional architectural solutions. DNA can exist in circular forms, in multiple independent molecules, and even inside organelles that were once free living cells. Circular DNA is one of the most widespread alternative structures. In circular DNA, the molecule forms a closed loop with no free ends. There are no telomeres to protect, no terminal sequences to shorten. The strand connects back onto itself, creating continuity. This arrangement eliminates the end replication problem seen in linear chromosomes, where a small fragment is lost during each round of copying. Bacteria provide the clearest example of circular genomes. Many bacterial species contain a single, large circular chromosome that floats within the cytoplasm rather than being enclosed inside a nucleus. The bacterium Escherichia coli has one circular chromosome containing several million base pairs. Despite its relative simplicity compared to human chromosomes, it carries all the information necessary for independent life. In bacteria, DNA is compacted through supercoiling candiena-binding proteins rather than histones arranged in nucleosomes. The overall logic of folding remains, but the structural details differ. Without a nuclear membrane, bacterial DNA occupies a region called the nucleoid. This space is not separated by a barrier. It is defined by organization rather than walls. Beyond the main bacterial chromosome, smaller circular DNA molecules often exist. These are called plasmids. A plasmid is an independent DNA loop that replicates separately from the main chromosome. Plasmids frequently carry genes that provide adaptive advantages, such as antibiotic resistance. Because plasmids can be transferred between bacterial cells, they contribute to rapid genetic change within populations. Plasmids behave almost like detachable modules. They are not essential for basic survival under normal conditions, but they can become critical in challenging environments. Their mobility allows genes to spread horizontally, meaning between organisms rather than strictly from parent to offspring. This mode of transfer differs from the vertical inheritance typical of human chromosomes. Circular DNA is not limited to bacteria. Inside human cells, mitochondria contain their own small circular genome. Mitochondria are organelles responsible for energy production, converting nutrients into usable cellular energy. Their DNA is separate from nuclear DNA and contains genes necessary for mitochondrial function. Unlike nuclear DNA, mitochondrial DNA is inherited almost exclusively from the mother. The presence of mitochondrial DNA reflects an ancient evolutionary event. The prevailing theory, strongly associated with Lynn Margulis, proposes that mitochondria originated as free-living bacteria that entered into a symbiotic relationship with early eukaryotic cells. Over time, most of their genes were transferred to the nucleus, but a small circular genome remained inside the organelle. A similar situation exists in plants. Chloroplasts, the organelles responsible for photosynthesis, also contain their own circular DNA. Chloroplast DNA encodes proteins essential for capturing light energy and converting it into chemical energy. Like mitochondria, chloroplasts are believed to have descended from ancient photosynthetic bacteria that became permanent residents within larger cells. These organelle genomes are much smaller than nuclear genomes, yet they operate with a degree of independence. They replicate separately and express their genes using machinery that retains bacterial features. Their existence shows that a single cell can contain multiple genetic compartments, each with distinct structural organization. When comparing genome sizes across species, another striking pattern appears. Genome size varies dramatically, often without a clear connection to organism complexity. This observation is known as the C-value paradox. The term C-value refers to the amount of DNA contained within a haploid nucleus. Some amphibians and plants possess genomes far larger than that of humans, despite not appearing more complex. This variation in genome size arises largely from differences in repetitive sequences, transposable elements, and non-coding DNA. Larger genomes do not necessarily contain more genes. Instead, They often contain expanded regions of repeated sequences, or structural DNA. The physical amount of DNA can increase without a proportional increase in functional coding regions. Genome size variation also influences cellular architecture. Cells with larger genomes often have larger nuclei. Larger nuclei can affect cell size, developmental timing, and metabolic rates. Thus, structural differences in DNA organization ripple outward into broader biological traits. Taken together, these alternative genome architectures demonstrate that there is no single blueprint for organizing genetic material. Linear chromosomes, circular genomes, plasmids, organelle DNA, and varying genome sizes all represent viable solutions. DNA adapts not only at the level of sequence, but also at the level of structure. Whether arranged in loops within bacteria, enclosed inside mitochondria, or folded into human chromosomes, the underlying molecule remains the same: sugar, phosphate, base. Yet the architecture shifts according to context. Life does not insist on one format. It selects the structure that best suits its environment, history, and scale. Before a cell divides, it must copy its DNA. Every letter, every sequence, every structural detail must be reproduced with extraordinary precision. A human cell contains billions of base pairs. Yet within a matter of hours, that entire genome is duplicated. This process is called replication, and it unfolds with quiet coordination. Replication does not begin randomly. It starts at specific regions along the DNA, known as origins of replication. From each origin, the double helix begins to open. The two strands separate, forming a structure called the replication fork. The replication fork resembles a widening Y-shaped junction, where the parental strands move apart and new strands begin to form alongside them. The separation of strands exposes the bases, making them available for copying. Because DNA strands are complementary, each original strand serves as a template for a new one. The pairing rules remain strict. Adenine pairs with thymine. Guanine pairs with cytosine. The structure itself guides the process. The central enzyme responsible for building the new strand is DNA polymerase. DNA polymerase adds nucleotides one by one, matching each new base to its complementary partner on the template strand. It links them together through chemical bonds, extending the growing chain in a precise direction. An important detail shapes the entire mechanism. DNA polymerase can only add nucleotides in one direction, from the 5' end toward the 3' end of the new strand. Because the two template strands run anti-parallel, this directional rule creates asymmetry at the replication fork. On one side of the fork, synthesis proceeds smoothly and continuously. This newly forming strand is called the leading strand. As the fork opens further, DNA polymerase moves steadily along, adding nucleotides without interruption. On the opposite side, synthesis is more complex. Because of the directional constraint, DNA polymerase cannot follow the fork continuously. Instead, it builds short segments of DNA in pieces. This strand is called the lagging strand. Each short segment is known as an Okazaki fragment, named after the scientists who first described them. These fragments are synthesized in the same 5-3 direction, but in small bursts as more of the template becomes available. Once a fragment is completed, the enzyme releases and begins another fragment slightly upstream. Later, these fragments are joined together to form a continuous strand. Before DNA polymerase can begin adding nucleotides, another enzyme must act first. DNA polymerase cannot start from nothing. It requires a short starting point called a primer. The enzyme primase creates this primer, a short stretch of RNA that provides the necessary starting end. Once the primer is in place, DNA polymerase extends from it. After the Okazaki fragments are formed, the RNA primers are removed and replaced with DNA. Then an enzyme called ligase seals the gaps between fragments. Ligase forms the final chemical bonds that connect the fragments into a seamless strand. Without ligase, the DNA would remain in disconnected pieces. Despite the speed of replication, accuracy remains remarkably high. DNA polymerase includes a proofreading function. As it adds nucleotides, it checks whether the correct base has been inserted. If an error occurs, the enzyme can remove the incorrect nucleotide and replace it with the correct one. This proofreading ability reduces the error rate to approximately 1 mistake per billion base pairs. The concept that each new DNA molecule contains one original strand and one newly synthesized strand was demonstrated in the Meselson-Stahl experiment. Matthew Meselson and Franklin Stahl used isotopes of nitrogen to track how DNA was distributed after replication. Their results showed that replication is semi-conservative, meaning each daughter molecule retains one parental strand and one new strand. This experiment provided strong confirmation of the double helix model's implications. Earlier biochemical studies, including those by Arthur Kornberg, identified and characterized DNA polymerase as the enzyme responsible for synthesis. These discoveries revealed that replication is not a spontaneous chemical reaction, but a carefully orchestrated enzymatic process. Replication does not occur at all times. It is tightly linked to the cell cycle, the ordered sequence of stages a cell passes through as it grows and divides. DNA replication occurs during a specific phase known as the S phase, short for synthesis phase. Only after replication is complete does the cell proceed toward division. This timing ensures that each daughter cell receives a full copy of the genome. The coordination of replication across billions of base pairs requires multiple replication forks operating simultaneously. In human cells, thousands of origins activate at once, creating a network of forks moving outward in parallel. This distributed approach allows the genome to be copied within a manageable time frame. At the molecular level, replication involves a complex assembly of proteins that stabilize the separated strands, prevent tangling, and coordinate synthesis. Yet the central principle remains elegant. Base pairing guides duplication. The replication fork advances, DNA polymerase extends, primers are laid down, fragments are sealed, and proofreading safeguards accuracy. The process resembles a highly disciplined copying system, not rushed but steady, capable of reproducing immense length with minimal error. Replication is not merely repetition, it is preservation. Each division carries forward the molecular continuity of life. The structure of DNA makes copying possible. The enzymes ensure it proceeds with fidelity. And each time a cell prepares to divide, this quiet choreography unfolds again. Two strands parting, two new strands forming, the genome renewing itself with remarkable precision. DNA is durable, but it is not invulnerable. Inside every cell, the genome is constantly exposed to chemical reactions, radiation, and mechanical stress. Damage is not rare. It is routine. What preserves stability is not the absence of harm, but the presence of repair. One common source of damage is ultraviolet light. UV radiation from the sun can alter the structure of DNA by affecting adjacent bases. In particular, two neighboring thymine bases may bond together abnormally, forming what is called a thymine dimer. A thymine dimer is a chemical link between adjacent thymine bases that distorts the shape of the double helix. This distortion interferes with normal copying and reading of DNA. The cell detects such distortions through surveillance proteins that scan the genome. When a thymine dimer is identified, a repair pathway called nucleotide excision repair is activated. Nucleotide excision repair removes a short segment of the damaged strand, including the abnormal bases. The gap is then filled in using the undamaged strand as a template, restoring the correct sequence. This pathway was studied extensively by Aziz Sankar, whose work clarified how cells recognize and remove UV-induced lesions. The repair process is systematic. It does not attempt to patch over the damage. It removes the affected section entirely and rebuilds it accurately. Not all damage involves large distortions. Sometimes a single base is chemically altered without dramatically bending the helix. For example, spontaneous chemical reactions can change cytosine into uracil. These subtle changes are corrected through base excision repair. In base excision repair, specialized enzymes detect the incorrect base, remove it, and replace it with the correct one. The surrounding backbone remains intact. These smaller corrections resemble careful editing at the level of a single letter. The overall structure remains stable, but individual errors are quietly removed. More severe damage can occur when both strands of DNA are broken. A double-strand break means the backbone of both strands has been severed. This is one of the most dangerous forms of DNA damage because there is no intact complementary strand at the breakpoint to guide repair. Cells address double-strand breaks through two main pathways. One is homologous recombination. Homologous recombination uses an intact, identical DNA sequence, often the sister chromatid formed during replication, as a template to accurately repair the break. The damaged region aligns with the undamaged copy, and the missing information is restored through precise copying. The second pathway is non-homologous end joining. In this process, the broken ends are brought back together and reattached without using a template. Non-homologous end joining is faster but less precise. Small insertions or deletions may occur at the repair site. It prioritizes continuity over perfection. These two strategies reflect a balance between accuracy and speed. When a template is available, the cell prefers careful reconstruction. When time is critical, it may accept minor imperfections to prevent larger instability. Despite these repair systems, not all damage is corrected. When an error escapes repair and becomes incorporated during replication, it becomes a mutation. A mutation is a permanent change in the DNA sequence. Mutations can involve a single base substitution, a small insertion, or a larger rearrangement. Many mutations are neutral, especially if they occur in non-coding regions or do not alter the function of a protein. Some are harmful. A few may confer advantages under certain conditions. Mutation is not inherently destructive. It is a natural byproduct of molecular imperfection. However, when mutations accumulate in genes that regulate cell growth or DNA repair itself, the consequences can be serious. Certain genes act as guardians of genomic stability. BRCA1 and BRCA2 are examples of such genes. These genes are involved in homologous recombination repair. When functioning properly, they help repair double-strand breaks with high accuracy. If BRCA1 or BRCA2 carry harmful mutations, the efficiency of repair decreases. Cells may rely more heavily on error-prone pathways. Over time, accumulated genetic damage can increase the risk of uncontrolled cell growth. The connection between impaired DNA repair and cancer risk illustrates how closely stability and health are linked at the molecular level. Yet even here, the system is layered. Cells possess checkpoints that monitor DNA integrity before division. If damage is detected, the cell cycle can pause, allowing repair to occur. If the damage is too severe, programmed cell death may be triggered. These safeguards limit the spread of errors. Across billions of cells, DNA is constantly being copied, damaged, inspected, and repaired. The process is not dramatic. It is continuous. Molecular machines scan along the strands, correcting distortions, replacing altered bases, sealing breaks. Stability in the genome does not arise from rigidity. It arises from vigilance. The double helix may bend, absorb radiation, or experience chemical shifts, but repair systems restore order before instability spreads. In this way, DNA is not only a carrier of information, but a system equipped to protect that information. Damage occurs, corrections follow, and through this quiet maintenance, the integrity of the genome is preserved across divisions, across tissues, across generations. DNA carries information in the form of sequences, but information alone does not act. For a gene to influence the life of a cell, its sequence must be read and converted into something functional. This process begins with transcription. Transcription is the step in which a segment of DNA is copied into RNA. RNA, or ribonucleic acid, is a molecular cousin of DNA that can carry instructions away from the genome. While DNA remains stored and protected, RNA serves as a temporary working copy. The central molecule that performs transcription is RNA polymerase. RNA polymerase binds to DNA and moves along one strand, building a complementary RNA strand by matching bases according to pairing rules. In RNA, thymine is replaced by uracil, but the principle remains consistent. Adenine pairs with uracil, and guanine pairs with cytosine. The enzyme links these RNA nucleotides together, forming a growing strand. Transcription does not begin randomly along the genome. It starts at specific DNA sequences known as promoters. A promoter is a region located upstream of a gene that signals where transcription should begin. It acts as a docking site for RNA polymerase and associated regulatory proteins. Within many promoters lies a short conserved sequence known as the Tata box. The Tata box contains a repeating pattern of thymine and adenine bases. Its structure helps position RN, a polymerase, precisely at the start site. It functions like a subtle marker embedded in the DNA sequence, indicating where reading should initiate. Once RNA polymerase is correctly positioned, it separates the DNA strands locally and begins synthesizing messenger RNA, or mRNA. Messenger RNA is the RNA molecule that carries the coding information from DNA to the cellular machinery that builds proteins. It represents a direct copy of the gene's template strand with the necessary chemical adjustments for RNA structure. In many organisms, particularly in eukaryotic cells such as human cells, the initial RNA transcript contains both coding and non-coding segments. These segments are called exons and introns. Exons are regions that will ultimately contribute to the final protein sequence. Introns are intervening sequences that are removed before translation. The removal of introns occurs through a process called splicing. Splicing is carried out by a complex molecular machine that recognizes the boundaries between introns and exons. The introns are cut out, and the exons are joined together into a continuous sequence. The result is a mature mRNA molecule that carries a refined version of the original transcript. This editing step allows flexibility. In some cases, alternative splicing occurs, meaning different combinations of exons can be joined together from the same gene. This enables a single gene to produce multiple protein variants. The underlying DNA sequence remains constant, yet its output can vary depending on how it is processed. The information carried by mRNA is organized in a 3-letter code. Every 3 nucleotides form a unit called a codon. Each codon corresponds to a specific amino acid, the building block of proteins. The mapping between codons and amino acids is known as the genetic code. The genetic code is nearly universal across living organisms. The same codon sequences specify the same amino acids in bacteria, plants, and humans. This universality reflects a shared evolutionary origin. Marshall Nirenberg played a key role in deciphering this code, demonstrating experimentally how specific nucleotide triplets correspond to particular amino acids. The concept that genes regulate cellular function in coordinated systems was explored by Francois, Jacob, and Jacques Renaud. Their work on gene regulation in bacteria revealed that genes are not simply turned on continuously. Instead, they respond to environmental signals and cellular needs. Although their studies focused on bacterial systems, the broader principle applies widely. DNA sequences function within networks. Transcription itself is tightly regulated. Not every gene is transcribed at the same time or in every cell. Some genes are active only in specific tissues. Others are activated only in response to signals. The promoter region integrates these influences, determining when RNA polymerase is allowed to proceed. At the molecular level, transcription is both precise and dynamic. RNA polymerase advances along the template strand, synthesizing RNA at a steady pace. As it moves, the DNA behind it reforms into its double helix. The reading window travels forward, while the rest of the genome remains structurally intact. Messenger RNA, once processed, exits the nucleus and travels to ribosomes, where the information it carries is translated into protein. But the essential step, the act of reading DNA into RNA, remains centered on the DNA sequence itself. RNA is a mediator. The blueprint resides in DNA. The transformation from sequence to expression illustrates how static information becomes active. DNA does not move. It does not leave its structural context. Yet through transcription, its sequences are interpreted and extended into functional molecules. Every time a cell needs a specific protein, a segment of DNA is accessed, read, copied into RNA, and prepared for translation. The process is orderly. Promoters guide the start. RNA polymerase builds the transcript. Introns are removed. Exons are joined. Codons define the amino acid sequence. The structure of DNA makes this possible. Its complementary strands allow accurate copying. Its promoters embed regulatory signals within the sequence itself. Its coding regions store instructions in triplet units that align with a universal code. From sequence to expression, the path is chemical, precise, and repeatable. The double helix that once appeared as a static spiral becomes a source of active output. Information held in molecular form becomes instructions carried forward. DNA remains the central reference. RNA serves as the transient messenger. And through transcription, the genome moves from quiet storage into functional presence, one gene at a time. Not every gene written in DNA is active at every moment. In fact, most genes remain silent in most cells. A neuron and a skin cell contain the same genome, yet they behave differently because different sets of genes are turned on or off. Regulation determines which parts of the sequence are used. One major regulatory mechanism is DNA methylation. DNA methylation involves the addition of a small chemical group, called a methyl group, to specific cytosine bases in the DNA sequence. This addition does not change the underlying letters of the code. Instead, it influences how easily that region can be read. When methyl groups accumulate near a gene's promoter region, transcription is often reduced. The chemical tag acts as a signal that discourages RNA polymerase from binding. It is less like erasing a sentence and more like placing a subtle mark beside it that says, "Not now." These methyl marks can be stable across cell divisions. When a cell divides, patterns of DNA methylation are often copied along with the DNA sequence itself. In this way, regulatory states can be inherited by daughter cells without altering the sequence. This form of inheritance is called epigenetic inheritance, meaning inheritance above or alongside the genetic code. Another layer of control involves histone modification. DNA is wrapped around histone proteins, forming nucleosomes. The tails of histone proteins can undergo chemical modifications, such as acetylation or methylation. These changes influence how tightly DNA is wrapped. When histones are acetylated, meaning an acetyl group is added, the chromatin structure often becomes more open. An open structure makes the DNA more accessible to transcription machinery. When certain methyl marks appear on histones, the effect can either increase or decrease gene activity, depending on the location. These modifications do not rewrite the DNA sequence. They adjust the physical context in which the sequence sits. If the genome is the script, histone modifications adjust the lighting and stage arrangement. Beyond local chemical marks, specific DNA regions called enhancers and silencers help regulate gene activity. Enhancers are sequences that increase the likelihood that a gene will be transcribed. Silencers decrease that likelihood. These regions can be located thousands of base pairs away from the genes they regulate. Enhancers function by binding regulatory proteins that help recruit RN, a polymerase, to the promoter. Silencers bind proteins that block or reduce transcription. Through the looping of DNA in three-dimensional space, enhancers can physically contact distant promoters. The genome folds in ways that bring these elements into proximity. Regulation, therefore, is not linear. It depends on spatial organization within the nucleus. Genes are influenced not only by nearby sequences, but by distant regions that come into contact through chromatin looping. Gene silencing can occur through combinations of DNA methylation, histone modification, and silencer activity. In some cases, entire regions of chromosomes become densely packed and largely inactive. This compact form limits access to transcriptional machinery. These regulatory systems allow cells to specialize. During development, certain genes are activated while others are permanently silenced. Conrad Waddington described development as a landscape in which cells move down branching paths toward distinct fates. His metaphor illustrated how gene regulation guides cells into stable identities without changing their DNA sequence. Epigenetic regulation also responds to environmental influences. Nutrition, stress, temperature, and chemical exposure can influence patterns of DNA methylation and histone modification. These environmental signals do not alter the genetic code directly, but they can shift how genes are expressed. Some of these changes are temporary. Others may persist long enough to influence long-term cellular behavior. In certain cases, epigenetic patterns can even pass through generations, though this is an area of ongoing research. Large-scale initiatives, such as the Human Epigenome Project, have sought to map these regulatory marks across different cell types. By identifying patterns of methylation and histone modification, researchers aim to understand how gene expression is coordinated at a systems level. Regulation adds nuance to the genome. A gene may be present, intact, and fully functional, yet silent in one tissue and active in another. The difference lies not in sequence, but in context. DNA methylation places chemical markers along the strand. Histone modifications alter the compactness of chromatin. Enhancers and silencers adjust transcription rates. Environmental signals shift regulatory states. Together, these layers form a dynamic system of control. The genome, then, is not a static instruction manual. It is a responsive network. Genes are activated, reduced, or silenced depending on cellular needs and external conditions. Through epigenetic control, DNA maintains continuity while allowing flexibility. The sequence remains stable across generations, but its expression adapts within each lifetime. Regulation ensures that the same genome can produce diverse cell types, respond to changing environments, and maintain balance across billions of cells. In this way, gene regulation transforms DNA from a fixed script into a controlled performance. The letters remain constant, the interpretation shifts, and within that shifting control lies much of the complexity of living systems. DNA is built for stability, but life depends on variation. If every genome were copied without change across generations, populations would remain genetically uniform. Diversity arises because small differences accumulate, reshuffle, and occasionally expand. These differences form the raw material for evolution. One of the most powerful generators of variation occurs during sexual reproduction. When reproductive cells are formed, homologous chromosomes pairs, inherited from each parent, align closely with one another. During this alignment, segments of DNA can be exchanged in a process known as crossing over. Crossing over occurs when matching regions break and rejoin in swapped positions. The result is a new combination of genetic material. Crossing over is a specific form of recombination. Recombination refers more broadly to the exchange or rearrangement of genetic segments between DNA molecules. Through recombination, alleles, different versions of a gene, are reshuffled into new pairings. This reshuffling increases diversity without altering the overall number of genes. Instead of inheriting entire chromosomes unchanged, offspring receive chromosomes that are mosaics of their parents' DNA. The genome becomes a patchwork of inherited segments. Recombination does not create entirely new sequences, but it reorganizes existing ones in novel combinations. At a smaller scale, variation can arise from single nucleotide changes. A single nucleotide polymorphism, often abbreviated as SNP, is a difference of one base at a specific position in the genome. For example, one individual may have a cytosine at a certain location, while another has a thymine. These single-letter differences are common across human populations. Most SNPs have little or no effect on function. Some occur in non-coding regions. Others may alter protein structure slightly. When SNPs occur in regulatory regions, they can influence how strongly a gene is expressed. Across billions of base pairs, even small variations can accumulate into meaningful differences. The Human Genome Project revealed that humans share the vast majority of their DNA sequence. Yet within the remaining fraction lies a rich landscape of SNPs and structural variations. These differences contribute to traits ranging from physical characteristics to disease susceptibility. Variation also emerges through gene duplication. Gene duplication occurs when a segment of DNA is copied more than once within the genome. The duplicate gene may initially perform the same function as the original. Over time, mutations can accumulate in one copy while the other maintains its original role. This process allows evolutionary experimentation. One copy preserves stability, the other explores new possibilities. Many gene families in complex organisms originated through duplication events. What begins as redundancy can become specialization. Transposons, sometimes called jumping genes, provide another source of diversity. Transposons are DNA sequences capable of moving from one location in the genome to another. They can insert themselves into new positions, sometimes disrupting genes or altering regulation. The existence of transposons was first identified through work inspired by earlier genetic principles established by Gregor Mendel, who described patterns of inheritance long before molecular mechanisms were understood. Transposons demonstrate that genomes are not entirely fixed maps. Certain sequences are mobile. When transposons insert near or within genes, they may change expression patterns. Sometimes the effect is harmful. Other times it introduces subtle variation that persists in populations. Over evolutionary time, transposons have contributed significantly to genome size and structure. Not all mutations alter function visibly. A silent mutation occurs when a change in DNA does not alter the resulting amino acid sequence of a protein. Because the genetic code is redundant, multiple codons can specify the same amino acid. A base substitution may change the codon, but leave the protein unchanged. Silent mutations still contribute to genetic diversity. They may affect how efficiently a protein is produced or influence RNA stability. Even when outward effects are minimal, they add to the background variation within populations. All of these mechanisms, crossing over, recombination, SNP formation, gene duplication, Transposition and mutation generate diversity at different scales. Some reshuffle large segments. Others alter single bases. Together, they create a dynamic genome. Variation alone does not determine which traits persist. Natural selection influences which variations increase in frequency over generations. If a genetic change enhances survival or reproduction under specific environmental conditions, it is more likely to spread. If it reduces fitness, it may diminish. Natural selection does not design, it filters. The diversity produced by mutation and recombination provides options. Environmental pressures determine which options remain. At the molecular level, diversity often appears small. A swapped segment here, a duplicated gene there, a single base altered. Yet across populations and generations, these small differences accumulate. The genome is not rewritten wholesale. It shifts gradually. Genetic diversity is therefore not a flaw in replication. It is a feature of biological systems. Copying mechanisms strive for accuracy, but slight variation is inevitable. Without it, adaptation would stall. The genome carries stability through faithful replication and flexibility through controlled variation. Crossing over rearranges parental contributions. SNPs introduce fine-scale differences. Duplications create room for innovation. Transposons add structural movement. Silent mutations accumulate quietly. Through these processes, populations maintain a balance between continuity and change. The DNA sequence is preserved, yet never identical across individuals. Each generation inherits both structure and subtle variation. And within that steady accumulation of difference lies the capacity for adaptation, resilience, and long-term evolution written not in dramatic shifts, but in countless small changes distributed across the genome. Most variation arises from slow shifts in DNA sequence. But in some organisms, adaptation takes less conventional paths. These systems do not rewrite the genetic code entirely. Instead, they adjust how it is interpreted, exchanged, protected, or inherited. One striking example appears in cephalopods, particularly in the species Octopus vulgaris. In these animals, especially within their nervous systems, RNA editing occurs at unusually high levels. RNA editing is a process in which the RNA transcript is chemically altered after it has been copied from DNA. The underlying DNA sequence remains unchanged, but the RNA instructions can be modified before being translated into protein. In octopus neurons, specific bases in RNA are altered in ways that change the resulting protein structure. This editing increases protein diversity without altering the genome itself. It allows the nervous system to fine-tune its molecular components. The DNA provides the template, but RNA editing adjusts the output. This strategy introduces flexibility at the level of expression rather than inheritance. The genome remains stable across generations, while the transcript can shift within the lifetime of the organism. It is a different form of adaptability, one that operates downstream of DNA. Another unusual mechanism is horizontal gene transfer. Unlike vertical inheritance, where genes pass from parent to offspring, horizontal gene transfer allows genetic material to move between unrelated organisms. This is common among bacteria. Through processes such as transformation, transduction, and conjugation, bacteria can acquire genes directly from their environment or from neighboring cells. A gene conferring antibiotic resistance, for example, can spread rapidly across a bacterial population without waiting for reproduction. Horizontal gene transfer alters the idea of a strictly branching evolutionary tree. In microbial communities, genes can move laterally. The genome becomes less like a fixed lineage and more like a network with intersecting paths. Some microscopic organisms push this flexibility even further. Belloid rotifers, tiny aquatic invertebrates, reproduce primarily through asexual means. In theory, long-term asexual reproduction should limit genetic diversity. Yet belloid rotifers display remarkable genetic variation. Research suggests that they incorporate foreign DNA from their environment into their own genomes. Fragments from bacteria, fungi, and plants have been detected within their DNA. Through horizontal acquisition and repair mechanisms, they maintain diversity without traditional sexual recombination. Extreme environments reveal another layer of genomic resilience. Tardigrades, sometimes called water bears, are microscopic organisms known for surviving intense radiation, desiccation, and temperature extremes. Their survival is linked in part to highly efficient DNA repair systems. When exposed to conditions that would fragment DNA in most organisms, tardigrades can enter a dormant state. Upon rehydration, their repair systems restore damaged DNA strands. The genome is not immune to damage, but it is capable of recovery. Stability here depends on repair capacity rather than avoidance of harm. In bacteria, adaptive defense systems provide yet another example of unusual genetic strategy. One of the most well-known is CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR functions as a form of molecular memory. When a bacterium survives a viral infection, it can incorporate fragments of viral DNA into its own genome at CRISPR loci. These fragments serve as records of past invaders. If the virus returns, the bacterium uses these stored sequences to recognize and cut the viral DNA. This system operates with precision. CRISPR-associated proteins, including Cas enzymes, target matching viral sequences and introduce breaks. The bacterium effectively uses genetic information from previous encounters to defend itself in the future. The molecular details of CRISPR were clarified by researchers such as Jennifer Doudna and Emmanuel Charpentier, who demonstrated how this bacterial defense system could be adapted for targeted gene editing. In its natural context, however, CRISPR is an immune strategy embedded in DNA itself. Mitochondrial inheritance offers a different form of exception. In most organisms, mitochondrial DNA is inherited almost exclusively from the mother. Unlike nuclear DNA, which recombines and mixes contributions from both parents, mitochondrial DNA follows a more direct lineage. Because mitochondrial genomes are circular and replicate independently, they accumulate mutations along maternal lines. This pattern allows scientists to trace ancestry across generations. It also illustrates that within a single cell, multiple genetic systems can operate under distinct inheritance rules. Across these examples, the genome is not merely a static archive. In octopuses, RNA editing reshapes output without changing sequence. In bacteria, horizontal gene transfer redistributes genes across populations. In rotifers, foreign DNA integrates into stable genomes. In tardigrades, repair systems counter extreme damage. In CRISPR, memory is stored within DNA to defend against viral threats. In mitochondria, inheritance follows a singular maternal path. These strategies do not replace the fundamental logic of DNA. Base pairing, replication, and repair still operate, but they demonstrate that the genome is adaptable not only through slow mutation, but through alternative modes of use. Life experiments not only with sequence, but with structure, exchange, memory, and recovery. The double helix remains the core molecule, yet its deployment varies widely across organisms. Through these exceptional systems, DNA reveals an expanded capacity. It is not confined to one pattern of inheritance or one mechanism of adaptation. It supports innovation while preserving continuity. And in that balance between constancy and creative strategy, genetic systems display a quiet flexibility, one that extends far beyond simple mutation and into the broader architecture of survival. DNA does not only function within a single lifetime, It extends across generations, linking individuals to ancestors they will never meet. Every genome carries traces of those who came before, arranged not as stories or images, but as sequences of nucleotides passed forward with each reproduction. One of the clearest lines of inheritance appears in mitochondrial DNA. Mitochondria, the energy-producing structures inside cells, contain their own small circular genome. Unlike nuclear DNA, which is inherited from both parents, mitochondrial DNA is passed down almost exclusively from the mother. This happens because the mitochondria in the fertilized egg originate primarily from the egg cell itself. Because mitochondrial DNA does not undergo the same recombination as nuclear DNA, it changes more gradually through accumulating mutations. Over many generations, small differences appear. These variations can be grouped into lineages known as haplogroups. By comparing mitochondrial sequences across populations, researchers can trace maternal ancestry far back in time. This method does not identify individual ancestors. Instead, it reveals branching patterns. Each mutation acts like a subtle marker along a path, indicating where lineages diverged. Through these patterns, scientists reconstruct migration routes and population expansions that occurred thousands of years ago. A parallel system exists in the Y chromosome. The Y chromosome is passed from father to son. Like mitochondrial DNA, large portions of the Y chromosome do not recombine extensively. Mutations accumulate slowly, forming identifiable paternal lineages. By studying Y chromosome variation, researchers can trace paternal ancestry and examine how male lineages have spread across regions. Combined with mitochondrial data, these markers provide complementary perspectives on human history. Beyond individual lineages lies the broader field of population genetics. Population genetics examines how genetic variation is distributed and changes within groups over time. It considers mutation rates, reproduction patterns, migration, and environmental pressures. One important concept in population genetics is genetic drift. Genetic drift refers to random changes in allele frequencies within a population. Even in the absence of natural selection, certain variants may become more or less common purely by chance. This effect is especially pronounced in small populations. If a small group becomes isolated, random fluctuations can significantly reshape its genetic makeup. Some alleles may disappear entirely. Others may become fixed, meaning every member carries them. Genetic drift operates quietly, without intention or direction. A related concept is the founder effect. The founder effect occurs when a new population is established by a small number of individuals. The genetic variation in this new group reflects only a subset of the original population's diversity. If the founders carry rare variants, those variants may become common in the descendant population. Over generations, these processes shape genetic landscapes. Migration introduces new variation. Isolation amplifies drift. Selection favors certain traits under specific conditions. Together, they create the patterns observed in present-day populations. Ancient DNA research has deepened our understanding of these processes. By extracting DNA from fossil remains, scientists can directly compare modern genomes with those of extinct populations. This approach has revealed that human ancestry is not a simple linear path. The work of Svante Pääbo and colleagues led to the sequencing of Neanderthal DNA. Their research demonstrated that modern humans and Neanderthals interbred tens of thousands of years ago. As a result, many people today carry small percentages of Neanderthal-derived DNA. These inherited segments are not symbolic. They are identifiable stretches of sequence embedded within the genome. Some influence immune function. Others have no clear effect. They persist as molecular traces of contact between ancient populations. The discovery of Neanderthal contributions changed the understanding of human evolution. Instead of complete replacement, there was admixture, meaning interbreeding between groups. Human ancestry is therefore braided rather than strictly branching. Deep ancestry is written not only in specific genes, but in patterns across the genome. Shared mutations indicate common origins. Divergent mutations signal separation. Over time, lineages split, migrate, reconnect, and diverge again. The pace of genetic change is measured through what is sometimes called a molecular clock. This concept assumes that mutations accumulate at relatively steady rates over long periods. By estimating mutation rates and counting differences between sequences, researchers approximate the timing of divergence events. Such estimates are not exact, they are probabilistic, yet they provide a framework for situating genetic changes within archaeological and fossil timelines. DNA becomes an additional line of evidence alongside physical remains. Through mitochondrial tracking, Y chromosome analysis, population modeling, and ancient DNA sequencing, the genome reveals continuity. It preserves fragments of distant histories within living individuals. Every person carries a mosaic of ancestral segments. Some trace back hundreds of generations. Others reflect more recent migrations. The genome does not store memory in narrative form. It stores variation accumulated through reproduction, drift, and contact. Inheritance ensures continuity, but it does not produce uniformity. Each generation reshuffles nuclear DNA through recombination. Mutations introduce subtle differences. Drift shifts frequencies. Selection shapes survival. Across deep time, these forces operate continuously. Populations expand and contract. Lineages split and merge. Genes move across regions. DNA persists through all of it, recording change through accumulated variation. The genome you carry is not an isolated creation. It is a continuation of countless replications stretching backward through time. Within its sequence lie patterns that connect present populations to ancient ancestors. Through careful comparison and analysis, those patterns reveal shared origins, events, and moments of contact long before written history. DNA, in this sense, extends beyond the cell. It forms a molecular bridge across generations, linking individuals to a broader human story embedded in sequence rather than stone. For most of history, DNA was invisible. Its structure was unknown, its sequence unreadable, its variations hidden inside cells. Today, the situation is different. The genome can be sequenced, compared, modeled, and in some cases, carefully edited. The shift has not been sudden, but it has been profound. One milestone in this transformation was the completion of the first human genome draft in the early 2000s, yet even that achievement left gaps. Certain regions, especially around centromeres and highly repetitive sequences, remained unresolved. In 2022, the Telomere-to-Telomere Consortium announced a more complete assembly, extending the sequence from one telomere to the other without missing segments. The letters were no longer scattered in fragments. The map became continuous. Having a complete reference genome does not mean that every function is understood. It means the entire sequence is accessible for study. It provides a stable coordinate system for future discoveries. As sequencing technology has advanced, the cost of reading an individual genome has dropped dramatically. What once required international collaboration and years of work can now be performed in days. This shift has made personalized genomic maps possible. A personalized genome is not a generic template. It reflects the unique sequence of an individual. These maps can identify variations associated with specific conditions. Early disease risk analysis often relies on statistical associations between certain genetic variants and increased probability of illness. For example, particular gene variants may correlate with higher likelihood of developing certain cancers or metabolic disorders. This type of analysis does not predict destiny. It estimates probability. A genetic risk marker increases likelihood. It does not guarantee outcome. Environment, lifestyle, and additional genetic factors all contribute. The genome provides part of the picture, not the entire story. Clinical gene editing has also entered careful experimentation. CRISPR-Cas9, originally discovered as a bacterial defense system, can be adapted to introduce precise changes in DNA sequences. CRISPR-Cas9 uses a guide RNA to target a specific DNA region and a Cas enzyme to cut the strand at that location. In clinical contexts, gene editing is being explored to correct mutations that cause inherited diseases. Some early treatments have focused on blood disorders where modified cells can be returned to the patient. These interventions are conducted under strict regulation and remain the subject of ethical oversight. Editing the genome is not the same as understanding it fully. Each gene functions within networks of interactions. A change in one region can have downstream effects. For this reason, gene editing applications proceed cautiously. Precision at the sequence level does not eliminate complexity at the systems level. Artificial intelligence has begun to assist in modeling genomic data. Machine learning systems analyze patterns across vast datasets, identifying correlations between genetic variants and biological outcomes. AI does not replace biological insight, but it accelerates pattern recognition. For example, AI models can predict how certain DNA variants may alter protein structure or gene regulation. They can also help interpret data from large population studies. As genomic databases expand, computational tools become essential for managing scale. Another frontier involves single-cell genomic analysis. Traditional sequencing often averages data across millions of cells. Single-cell techniques allow researchers to examine genetic expression patterns in individual cells. This reveals variation within tissues that would otherwise remain hidden. In complex organs such as the brain or immune system, single-cell analysis can identify distinct cellular subtypes and track how gene expression shifts during development or disease. The genome remains the same in most cells, but its activity varies. Single-cell genomics sharpens the resolution. Beyond medicine, DNA is being explored as a medium for data storage. DNA data storage involves encoding digital information text, images, or code into sequences of synthetic DNA. Because DNA is extremely dense as an information carrier, small volumes can store large amounts of data. Experimental systems have successfully encoded books, images, and other files into DNA strands and later retrieved them by sequencing. While not yet practical for everyday use, the concept demonstrates DNA's stability and density as a storage medium. It transforms a biological molecule into an archival material. The emerging genomic era is characterized by integration. Sequencing technologies, computational modeling, molecular editing, and single-cell analysis converge into a broader framework. The genome is no longer viewed solely as a fixed code, but as a dynamic system interacting with environment and development. Yet progress remains incremental. Each advance clarifies one layer while revealing additional complexity. Mapping a genome does not automatically explain regulatory networks. Editing a gene does not guarantee predictable outcomes across tissues. In this landscape, the language of possibility is appropriate. Personalized genomic maps may support preventive medicine. Gene editing may expand therapeutic options. AI may refine interpretation. DNA data storage may offer long-term archival solutions. Each development carries potential, not certainty. What remains constant is the molecule itself. The same double helix described decades ago now sits at the center of computational models, clinical trials, and bioengineering laboratories. Its structure has not changed. Our ability to read and interact with it has. The future of genomics is unlikely to be defined by a single breakthrough. It will likely emerge through steady refinement, better sequencing, more accurate models, careful applications, ethical oversight. Large transformations may occur, but they will be built on incremental understanding. In this sense, the genomic era extends the same principles seen throughout DNA's story. Stability paired with flexibility, precision balanced with caution, innovation guided by structure. The genome carries history across generations. It also serves as a platform for exploration. As technologies evolve, the molecule remains the reference point, a stable thread running through biological research. The double helix once revealed how information could be stored. Today, it offers insight into how information might be interpreted, modeled, and in limited contexts, modified. The arc from discovery to application has been gradual. And as we look forward, the vision is not of dramatic rewriting, but of careful reading. The genome is vast, layered, and interconnected. Its future lies not in spectacle, but in sustained attention, sequence by sequence, cell by cell, generation by generation.